and welcome to Spotlight on Antibodies, a podcast from BioWintron, where we bring you the latest antibody industry trends. This month in March, we'll highlight antibody allostery, trends in developability, and anti-IgE therapeutics. Let's take a look at antibody allostery, an area that's been gaining attention as new structural and computational studies challenge some long-standing assumptions about how antibodies function. Traditionally, antibodies have been described as modular molecules, with the variable or Fab region responsible for antigen binding, and the constant or Fc region mediating effector functions. These domains were often treated as largely independent, but over time, that simplified view became harder to maintain. A growing body of evidence now suggests that there may be communication between these regions, what researchers refer to as antibody allostery. This means that changes in one part of the molecule may influence behavior in another, even across relatively long distances within the structure. However, what's emerging is a more nuanced picture where different types of coupling can occur under specific conditions. For example, some studies distinguish between conformational allostery, where structural changes propagate through the protein, and other effects like glycosylation-driven structural variation or multivalent interactions between antibodies and clustered antigens. These distinctions matter because not all observed changes in antibody behavior necessarily reflect direct signaling between domains. More recent work has focused on how context shapes these interactions. Computational analyses suggest that perturbations, whether from ligand binding, sequence variation, or post-translational modifications, like glycosylation, can travel through domain interfaces and hinge regions. In some models, binding near the CH1-CH2 interface has even been proposed to influence antigen binding at the Fab region. But much of this evidence remains computational, and experimental validation is still limited. At the same time, the concept of allostery is expanding beyond the antibody itself. Increasingly, antibodies are being studied not just as binding molecules, but as modulators of their targets. In one recent example, involving the epidermal growth factor receptor, certain antibody formats were shown to induce long-range changes within the receptor, affecting downstream signaling pathways like ERK and AKT. Interestingly, these functional effects didn't always correlate directly with binding affinity or receptor internalization, suggesting that how an antibody engages its target can influence signaling outcomes in more complex ways. Similar observations have been reported in other systems, such as bacterial adhesins, where antibodies can interfere with ligand binding through multiple mechanisms, including conformational stabilization and steric effects. In some cases, these interactions appear consistent with an allosteric mode of inhibition, although the exact mechanisms can be difficult to distinguish. Another important factor in all of this is glycosylation. Variations in Fc glycoforms are known to affect antibody flexibility and effector function, but there's also emerging evidence that glycans can play more direct roles. In certain antibodies, glycans within the binding region can contribute to antigen recognition itself, acting almost like structural components rather than passive modifications. So overall, what we're seeing is a shift away from viewing antibodies as strictly modular toward a more conditionally integrated model. For antibody engineering, this means that factors like epitope selection, molecular format, and glycosylation may influence function in ways that aren't always obvious from binding data alone. Following that discussion on antibody behavior, we're seeing a growing focus on antibody developability. Essentially, how well a candidate antibody can move from discovery into a manufacturable, stable, therapeutic product. As monoclonal antibodies continue to expand across oncology, immunology, and infectious disease, the scale of discovery has changed significantly. New platforms can now generate hundreds or even thousands of candidates against a single target. But with that scale comes a new challenge. Not every antibody that binds well will perform well during development. Developability refers to a set of molecular properties that influence whether an antibody can be efficiently produced, formulated, and maintained as a consistent product. These include factors like structural stability, aggregation tendency, hydrophobicity, charge distribution, and nonspecific interactions. Importantly, these properties don't always correlate with antigen binding or functional potency, which means they can be overlooked if the focus is only on biological activity. One of the clearest trends is that developability assessment is moving earlier in the discovery process. Historically, lead selection was driven primarily by binding affinity and functional assays, with biophysical characterization coming later. But experience has shown that many downstream issues, like aggregation during purification or instability during storage, are rooted in intrinsic molecular features that aren't captured in those early functional screens. As a result, many groups are now incorporating developability screening alongside biological evaluation from the beginning. Rather than acting as a strict filter, these assessments provide additional context when comparing candidates with similar activity, helping teams identify potential risks earlier. Another important shift is the move towards multi-parameter profiling. Antibody behavior is rarely determined by a single property. Instead, it reflects a combination of structural and physicochemical characteristics. So instead of relying on one assay, like thermal stability, researchers use panels of assays that evaluate multiple attributes at once. This more integrated approach allows for a clearer understanding of how an antibody might behave under real-world manufacturing and formulation conditions, especially when dealing with large candidate pools. This leads into another key trend, the rise of high-throughput screening. With modern discovery methods generating large numbers of candidates, traditional analytical techniques can become a bottleneck. In response, newer methods are being developed that use smaller sample volumes, plate-based formats, and parallel workflows to rapidly assess developability across many molecules at once. At the same time, machine learning is beginning to play a larger role. Predictive models can now estimate properties like aggregation risk or chromatographic behavior directly from antibody sequences or structural models. These tools are not replacing experimental data, but they are helping prioritize candidates before lab testing begins. And importantly, they depend on high-quality experimental datasets for training, which creates a feedback loop between computational prediction and experimental validation. Finally, there's a shift toward thinking about developability as part of antibody design itself. While antigen binding is largely determined by the CDR regions, the surrounding framework contributes to overall molecular behavior. Adjusting framework residues or selecting different germline frameworks can sometimes improve properties like solubility or stability without compromising binding. So overall, developability is no longer just a downstream consideration, it's integrated across discovery, screening, and design, helping teams make more informed decisions earlier in the pipeline. Building on those trends, our last segment shifts to a therapeutic area where these antibody engineering and developability considerations are actively being applied. Immunoglobulin E, or IgE, is most commonly associated with allergic disease. It binds to receptors on immune cells like mast cells and basophils, and when allergens are introduced, this interaction triggers inflammatory responses. Therapies such as omalizumab were developed to interrupt this pathway by binding circulating IgE and preventing receptor engagement. But more recent research suggests that IgE's role extends beyond these immediate allergic responses. For example, IgE also interacts with dendritic cells, which are responsible for presenting antigens to T cells and shaping downstream immune activity. Through these interactions, IgE can enhance antigen presentation, promote type 2 immune responses, and influence cytokine production, including increasing inflammatory mediators like IL-6. These effects appear to influence the balance between inflammatory and regulatory pathways. One area of interest is the impact on regulatory T cells, or Tregs, which help maintain immune tolerance. Some studies suggest that IgE signaling may impair Treg function, potentially contributing to sustained inflammation and allergic disease. This broader view of IgE biology has led to renewed interest in how anti-IgE therapies might affect immune regulation more generally. In addition to reducing activation of mast cells and basophils, anti-IgE treatment has been associated with decreased expression of the high-affinity IgE receptor, Fc-εRI, on immune cells. There is also evidence that dendritic cells may shift towards a less inflammatory, more tolerogenic profile, and in some cases, increases in Treg populations have been observed following treatment. Looking ahead, there is increasing activity around next-generation approaches that go beyond simply neutralizing circulating IgE. Some investigational therapies are designed to more extensively reduce IgE levels, including in patients with very high baseline concentrations. Others aim to inhibit IgE production upstream, targeting the processes that generate IgE in the first place. There are also strategies focused on the level of effector cells. Instead of only targeting free IgE, these approaches attempt to interfere with IgE signaling directly at the cell surface, where receptor-bound IgE drives immune activation. Early-stage programs, including a phase 1 candidate from Poplar Therapeutics, are exploring multifunctional mechanisms that combine IgE neutralization with suppression of production. Meanwhile, recent industry activity, such as acquisition efforts by Novartis, points to growing interest in these alternative strategies. Overall, what's emerging is a more complex picture of the IgE axis. Rather than acting solely as a trigger for allergic reactions, IgE appears to play a broader role in immune regulation. And while current therapies are effective for many patients, new approaches are being developed to more comprehensively target these pathways. And that's all for this month's Antibody Industry Trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.